The next topic in Chapter 3 is cell division. There are two types of cell division, mitotic cell division and meiotic cell division. Mitotic cell division creates two new cells from an identical cell. What's called a parent cell creates two identical daughter cells, and occurs when cells are being replaced during an embryo's growth or after damage. Meiotic cell division is developing cells that have half the number of the original chromosomes, where one cell creates two cells, and those two cells then create four total cells. This occurs when sex cells are formed, preparing for reproduction. Mitotic cell division is broken up into three processes: replication of chromosomes, otherwise known as DNA replication or DNA synthesis; the second one, mitosis, also known as nuclear division; and the third, cytokinesis, which is also known as division of the cytoplasm or cytoplasmic division. These processes are all a part of what's known as the cell cycle, which follows the divisions of the cell. Most of the cell cycle is in a stage we call interphase, or what was once thought of the stage between the cell division, which we now know is where a lot of the cell growth occurs. In what's called growth phase 1 or G1. This is also when we have DNA replication or DNA synthesis, otherwise known as S phase. S being for DNA synthesis, but again known as DNA replication or the copying of chromosomes. And G2 phase or growth phase 2, the growth that takes place prior to the actual cell division event, which we call nuclear and cytoplasmic division, or otherwise known as mitosis, where that one cell creates two identical new cells. After G1 phase, which is the growth phase where cells increase in size and volume and numbers of organelles, we have S phase, where DNA replication occurs and where a single chromosome creates two new chromosomes. And in that process we have what's known as unwinding, pairing, and joining, or a single DNA molecule gets copied into two DNA molecules, what's known as a chromatid becomes what's known as a sister chromatid. A single chromosome becomes a copy chromosome. In unwinding, the DNA double helix with its complementary nitrogen-base pairs, adenine and thymine and cytosine and guanine, break apart from one another or unzip between the nitrogen bases and the hydrogen bonds that hold them together, such that the double-stranded molecule forms single strands. The next process is known as pairing. When the free nitrogen base pairs that are found inside the cytoplasm will pair up with the available nitrogen bases on the exposed strand, such that a free thymine nitrogen base will bond with an adenine, and a free guanine nitrogen base will bond with a cytosine. What follows is joining, where those new nitrogen bases bond together and we reform a double-stranded molecule. This time 2 nitrogen base strands, not just 1. To recap DNA replication, a single chromosome of DNA unwinds to create 2 single strands, pairs with free nucleotides and nitrogen bases, and then joins. So as a chromatid or single chromosome becomes a sister chromatid or copied chromosome. And this copied chromosome has 2 identical double strands that are held together temporarily by a structure known as a centromere. Preparing to be divided during the process of cell division and mitosis into 2 new cells. So the process we just described happens in a cell in the stage called interphase, which looks like just a typical cell, where the nucleus in the middle went from having single chromosomes or chromatids into sister chromatids. And all the chromosomes within that cell have been copied. And the state in which these chromosomes are in this nucleus is what's called chromatin, or unraveled chromosomes. So the nuclear membrane is still intact around this nucleus. And out in the cytoplasm we have what are known as the centrosome, which is made up of a pair of centrioles that are going to be used later to divide those sister chromatids and help divide the cell. As the cell then enters into mitosis, or the actual cell division, the first stage is known as prophase. In prophase, that nuclear membrane starts to disperse, releasing those chromosomes as the chromosomes begin to condense or coil up. So what were once long stringy chromosomes in what we said was the state called chromatin, we now have condensed chromosomes, which are still in that copied state we call sister chromatids. That centrosome separates such that we have centrioles that go to opposite ends of the cell. And these networks of protein fibers start to release individual protein fibers known as spindles. These spindles or spindle fibers will begin to connect to these chromosomes and align them within the cell. Once those spindle fibers have distributed those chromosomes across the center of the cell, we're in a stage known as metaphase. So all the chromosomes within one cell are lined up across the center of the cell, known as the equator of the cell, or the metaphase plate. And those spindle fibers are connected to those sister chromatids at the center of the cell, the center of the chromosome, known as the centromere. The next stage begins as these spindle fibers pull those copied chromosomes apart from one another and break that centromere that connects them. And then we transition to the next stage we call anaphase. In anaphase, those sister chromatids are broken into chromatids. And as identical copies, each chromatid then will be carried to the opposite end of the cell. The last stage of mitosis is known as telophase. And this is when those chromatids, or those single copies of DNA, now make it to each end of the cell. They begin to unravel, and a new nuclear membrane forms around it to create two new nuclei. Once this is accomplished, then we see the splitting off of the cell. And that division of the cytoplasm we called cytokinesis. So the cell membrane now separates two cytoplasms and two nuclei, and essentially creates two new cells. These points in which the cells pinch off from one another is known as a cleavage furrow. To summarize this process, as a result, a single cell that was in interphase and had copied its chromosomes in S phase in the process called DNA replication, then splits into two new cells through the 4 stages of mitosis: prophase, metaphase, anaphase, and finally telophase, which ends with that splitting of the cytoplasm known as cytokinesis. And one parent cell creates 2 new identical daughter cells and completes the cell cycle.